Good morning, dear students. Today we will do the last part of absorption by roots. And here are some experiments based on absorption of water. So let's begin. So you have three different experimental setups here. The first two test tubes X and Y. Both of them are filled with water and a layer of oil has been added on top of both of these. A small seedling has been inserted in the test tube Y. You leave this entire experimental setup out in the open for a couple of hours. After some time, you will notice that the water in the test tube Y falls down, whereas it remains untouched in the test tube X. This happens because the leaves of the seedling are continuously losing water by transpiration, and the roots are continuously absorbing water to compensate for the lost water. Due to this, the level of water falls down. But in test tube X, nothing happens. There is no evaporative loss of water from the surface because of the layer of oil. So the test tube X acts as a control setup. Now look at the experimental setup in containers A, B, and C. Again, you have the seedling in A and C. In case of B, the leaves of the seedling have been removed. Water has been added in all three containers. A layer of oil has been added on top to prevent evaporative loss of water from the surface. Now, the entire experimental setup is left as such for an hour or so. In the container C, a source of additional light and heat, that is a bulb, is added. So we notice that the level of water falls down rapidly in C, then in A, and then in B, almost it remains untouched. Why does this happen? Because the rate of transpiration is maximum in case of the plant or the seedling in case of C. Why is this so? Because there is an additional source of light and a little bit of heat. This will increase the rate of transpiration, so the roots will absorb water faster and the level of water in container C will go down faster. If on the other hand you compare container A, the leaves are going to transpire water and the water will be absorbed from the container below, due to which the level of water will fall down in A as well. But the level of water will fall down in C much faster than it will in A. And in case of container B, the level of water will fall down almost negligibly because there is some transpiration which will take place from this this remaining structure of the plant, that is the stem. It will also transpire, but the rate of transpiration will be much less than it would if it were having any leaves. The third experimental setup simply is used to demonstrate that the roots absorb water and transpiration takes place from the leaves. So here we have a test tube containing water, having a thin layer of oil above, and the seedling is placed, inserted inside. After some time, we will notice that the level of water has fallen down. Why has it fallen down? Because the leaves have transpired water and the roots have taken in more water to compensate for the loss of water. In doing so, the level of water falls down. No water was lost from the surface. To ensure this, we have a thin layer of oil above. So this means that the water lost was absorbed by the roots to enable the leaves to transpire. So these three experimental setups show that roots absorb water. Okay, the next experimental setup tells you that the xylem in the root absorbs water. Now, you have the first experimental setup, a balsam plant, which has been kept in a beaker full of eosin dye. When you place a balsam plant in eosin dye, the roots, as you can see, have been removed. So the twig has been placed here. The xylem will absorb water because the leaves are going to transpire. There will be a suction force created to enable ascent of sap, and a little bit of water will be absorbed by the twig as long as it doesn't die. Now, after an hour, when you examine one microscopic slide of the stem under a microscope, you will notice a thin slice of this stem is observed under the microscope, you will notice that the xylem part is stained red, indicating that the xylem was the structure through which the water rose up during ascent of sap. Now here you have two experimental setups. In one, the part of phloem and bark has been removed from a piece of stem with the leaves intact. In the other, the part of xylem has been removed, a hole has been drilled and part of xylem has been removed, leaving the leaves intact. There are no roots, so this is only going to survive for some time. So this is a part of a twig. Now we notice after an hour or so that the leaves which were placed in the plant part having the xylem intact, those leaves remain turgid, whereas the ones which were attached to the twig where the xylem has been removed, those leaves wilt. This happens because the xylem provides the leaves with water. But here there is no xylem, so the water is not reaching the leaves, and in turn, the leaves are wilting. Now, here are various experimental setups to show that water is conducted upwards in a plant through xylem. So if you take celery leaves, or if you take any flower which is whitish in color, and you submerge it in a container full of colored water, the entire plant structure will acquire a tinge of the color in which it is immersed. Why does this acquire that color? Because the xylem is allowing for ascent of sap, and in doing so, the color travels up through the entire length of the plant, as you can see here. So the xylem part gets colored due to the section in which the celery or the colored flower, the flower was kept, and the xylem will be continuously colored, indicating that the upward movement of water takes place across xylem. So all these experimental setups are basically meant to tell you that the upward movement of water and minerals takes place through the xylem. Now, next you're having the ringing or the girdling experiment. Now, what is the ringing or the girdling experiment? In any plant, when we perform an experiment in the lab, we use a twig in which the roots have been removed. So here, a small ring of bark and phloem is removed from the plant. The entire plant setup is kept in a beaker or a container full of water for some time. So afterwards, we notice that the leaves remain erect. That means the leaves are turgid still. They are able to photosynthesize and they are able to prepare food. That food comes down and accumulates here because there is no further channel of food since we have removed the phloem part. The food cannot go down. It remains here. So this portion swells up. And the portion below gets deprived of any nutrition, any food. So this part of the stem slowly and slowly starts dying out. This is called as girdling. So this is a common method used in increasing the sugar content of certain plants. But otherwise, girdling, as you can see in this case, the stem has swollen up with swollen food. With stored food. Here also the stem swells up with stored food. Otherwise, girdling is used as a method to cut the trees. So once this portion is swollen up with the stored food, the stem below does not receive any nutrition. Okay, when the stem below will not receive any nutrition, the roots and the lower part of the stem slowly and slowly will stop growing and will die out. So the tree can be uprooted. This is a method of removal of trees in certain areas where it's not easy to fall down the entire tree or it would require a lot of effort. So this takes time. It's a slow process, but yes, it kills the tree. So this is a method in which we prevent the sugar content of the tree, of the leaves, to reach the lower stem, and hence it's used to remove the trees. So it's called girdling. Now, next you are having the forces which contribute in the ascent of sap. So we have 5 forces basically. Number 1 is the root pressure. Cell-to-cell osmosis creates an upward force by the roots. That upward force is called as root pressure. This force will enable the rise of water up to great heights. The second is capillarity. The thin diameter of the xylem vessels will enable the upward movement of water to greater heights. This is called as capillarity. Then there is transpiration pull. The leaves are continuously transpiring water, losing water as water vapor to the aerial part, to the atmosphere. In fact, all aerial parts of the plant, whether it's the leaf or whether it's the stem, they transpire. So when they transpire, they create an upward pull, which is called as transpirational pull. Then there is adhesion. Adhesion means the force of attraction between water molecule and the xylem tubes. So the water sticks to the xylem, and in this, it creates a force which we call as adhesion. Water molecules also stick to each other, that is called as cohesion. So the two water molecules form a channel sticking to each other and yet sticking to the sides of the xylem tubes in which they are found. So here you are being able to study the capillarity. If you have a tube of a larger diameter or if you have a narrow capillary, you can easily see water rises to a greater height in a capillary. That is why xylem vessels and xylem tubes, they are best suited for absorption of water because of their narrow capillary structure. So these are the various forces. Roots will absorb water, which will create a root pressure. The narrow capillary structure of the xylem will further help in ascent of sap. The water lost by transpiration will create a suction pressure or a transpiration pull. All of this will help in ascent of sap. Now, as you can see here, the water molecules, when they stick to the sides of the xylem vessels, that is called as adhesion. And when the water molecules attract each other, that is called as cohesion, as you can very clearly see in this diagram. When the water molecules attract to each other, so they will rise up as a column, that's cohesion. And when water molecules are attracted or attached to the xylem vessel, it is called as cohesion. Both of these will prevent the water molecules from falling back from where it is coming, and then the water will be rising upwards only. Remember, ascent of sap is against the gravitational pull. So that means something has to keep it going up. So all these forces, whether it is root pressure, whether it is capillarity, whether it is transpirational pull, or whether it is cohesion and adhesion, will ensure that the upward movement of sap continues undisturbed. So here, once water is absorbed by the roots, it creates a root pressure, and due to capillarity and the water lost by the transpiration, upward movement of water takes place even in the tallest of trees. If downward movement of sap has to take place, that is pretty simple. It takes place through phloem and it takes place due to gravity. So everything has to go down. It will be attracted by gravity. So these are the forces which will help in the ascent of sap and the downward movement of sap. So downward movement is through gravity and upward movement is through root pressure, capillarity, transpirational pull, and adhesion and cohesion. So with this, we finish the last part of absorption by roots. Thank you very much and God bless you all.